Thank you very much. I think I introduced some of the corporate messagings and vision in the first part of my presentation, was a thematic presentation. So I'm going to be brief on that part. As you can see, our company was established in 2017, and we have about 69 employees working at our company. As you know, that our focus areas are immunotherapy against cancers, and also allergy therapies and other immuno diseases. And we identify clinical unmet needs and we put them on smart platforms to develop them further. And there is a GI 101 project, and that technological transfer happened in 2019. And we also have joint research partner, Yonsei University's Kyungchul Choi, professor at Yonsei University, and we were chosen as top 100 national R&D performances in 2020. And when you visit our company, I would like to show off that we have to create best facilities, best labs, so that researchers can focus on their study subject. That's his vision and philosophy. So we can really show you that we have state-of-the-art technology, devices, and equipment, and our researchers utilize these options and facilities to focus on their research. And Myung-Ho Chang, a CEO, was a scientific advisor at Genexine. He was a professor at Osaka University, and he also worked as a principal investigator at IBS, and then worked at Genexine, and then became CSO at Progen, and then decided to start his own company called GI Innovation. And another CEO, as you know, Suyeon Nam, is quite popular. He got MD and PhD at Yonsei University, worked as a medical director at Roche Korea, and then worked at CSO for multiple years at Yuhan Corporation. And then she achieved many milestone breakthrough moments throughout her career, and now she's serving as CEO, CMO, and CBO. Recently, we are preparing to go public, and as for that, we decided to hire Junho Hong, who was the first CEO of Interpark Bioconvergence Corporation. We put in a lot of efforts to hire him and convince him to join our team. We also have Yongyu Cho, Vice President at Biologics Incorporation. And there are other team members who are experienced in multiple areas and fields of business and medical. And this is how we are operating our company. Because we are a small sized venture company, we cannot produce proteins ourselves. So in the very initial stage of our company, we decided to have partnership with CDMOs, and Samsung Biologics have been providing GI 101 protein. They have been very helpful for us, and we're co-developing very smoothly. And for allergen therapeutic, which is GI 301, we are partnered with Binex. And then for GI Smart Tab TM platform, we also have antibody targeting technology. And for the adoption of the technology, we worked as investor too, but also working with biologics. And for GI platform technology, which is our representative technologies, we can increase efficacy and we can also bring the cost down. There are Smart Clevo and Smart Selects technologies that we have. And for Smart Clego, it is basically encouraging fast cloning by using Lego-like block integration. This is why we call the technology Smart C Lego. However, moving on to Smart Selects, this is what is important is API, and what is important is the bridge role. Linker or hinges could be very important because we have to create a stable environment so that proteins can actually become effective, and that is why we decided to call this SELEX. And as you can see, we have GI 101 and GI 301, and there are other pipelines underway. And for 101, this is being used as a immuno-oncology treatment, and we have transferred this technology to a Chinese corporation, and then currently we are granted with Keynote 101 and now going through clinical stage. For GI 301, we outlicensed to Yuhan, and because it's Japan based, we are currently considering another round of technological transfer to Japanese organizations as well. So we have patents on both GI 101 and GI 301. Because I already covered GI 101, I'm going to be very brief on introducing GI 101. There's FC and there's CD 80 and then FC and then interlunker 2 variants. What is very important is that CTLA4 trap is composed of ECD, endogenous CD80, and the most promising medicine is now sold in the market, but the problem is that it has high side effects, and patients have a hard time to be administered with this product for a long time. So we decided to design a stabler product, and I think this is interest of many other global pharmaceutical companies as well. NK Cells, NKTR signed a global deal with a size of 4 trillion won, and if you look at that specific product, it's failing to increase the temperature up to a specific amount, a point, and we are trying to make it safer by not having to deal with the temperature again. And now going forward to the actual mechanism of GI 101, we it basically interrupts CD80 and CTLA4 interaction with IL-2 variants to activate and proliferate T cells and NK cells. And then as a result, just to be sharing briefly, what matters is that you have to block CTL4 as much as possible. And this is already available in commercially, so we have been checking regularly, and you can see that it is encouraging T cell activation by CTR stimulation, and it can re-encourage the stimulation very effectively. So maybe there would be a question whether you actually saw this response and reaction, and compared to the conventional RL IL2 CD8 IFN, GL101 has a better efficacy and better reaction, and it also contains a higher volume of cytokines as well. So now going back to the primitive data, when you administer GI 101 twice every 3 weeks, you can see that both T cells and NK cells have been activated. And what is better is that if you think about conventional cytokine, when you do the second dosing, the response drops significantly, and then there is a slope and a slowdown. However, you can see that our magnitude has been retained, amount of magnitude has been repeated. And whenever you give again and again, the magnitude goes back up to the previous point. And if you think about over here, this assay was created by Professor Choi at Yonsei University. When you actually give a GI 101, we have to see whether TIL, the tumor infiltrating lymphocytes, are going in or not. If you put an isotype control, there's not much being seen, whereas MGI 101, you can see that TIL is being shown, and this means that it is effective when it comes to cancer fighting, cancer treatment. And because we can activate multiple points in a cancer treatment, you can combine other therapies, and we wanted to make sure the results were guaranteed, and we have linked this into clinical study design. So representative data sheets over here is humanized mice. This humanized mice did not have immunity, and then we give a PVMC of human blood, and so that the mice could share the same immunity environment. And then we put breast cancer, TMBC, cancer cells in order to carry out experiments, and we give Cambridge Marketuda at the same time, and then you can see that when we put GI101 alone without the Kembruda, the effect was good enough. And when you did combined therapy with Keytruda, you can see that the efficacy went up as well. And it also showed that it has a memory feature as well at the end. And VEGF inhibitor or reduced therapy could be combined with GI101, and it showed a synergetic effect for sure. So, with utilizing these results, we are now currently designing our clinical studies, and we were granted with PKOT, and we are going to carry out joint research with them. And as I said, we did technological transfer to Simcere. It's one of the Chinese innovative companies approved by the Chinese government, and about 900 billion won size of a joint technological transfer was carried out. And by utilizing the clinical results in the future, I think the transfer growth is going to be expected. And another type of technological transfer was related to Yuhan, so we did not do separate IR for this. And for Japan, it is our strategy to exclude Japan because we have to carry out a specific clinical study separately. And it seems that their regional patient group is different from other countries, so we're going to we carried out a technological transfer with a different approach, and joint clinical studies are going to be carried out smoothly, I believe. Sorry, I showed this wanted to show this earlier, but couldn't because there was some screen error. Now I have to talk about next pipelines. Now, after GI 101, we have other pipelines underway. For there's N series, there's the 2 series, and there's 3 disease, which is about metabolic diseases. And for GI10N series, not only CD8 T cells go up, but other NK cells are successfully increased in number. We are going to be specifically targeting NK cells by using GI10N, and these types of medicines, we always have to think about side effects. For example, this is also the monkey data, and you can see the body mass and weight has been decreasing consistently, but our medicine, it doesn't show any weight loss, and it is indirectly proving that it's safe enough. And then another data that is presented is global pharmaceutical companies are developing NK cell activators and T cell activators. Compared to their open public clinical data, I think our NK cell activator is going to be better. Because this is monkey data, I believe that this results can be successfully transferred to human body.